In this section, we're going to discuss feature prediction through PICRUSt1 and PICRUSt2. The high cost effectiveness of 16S amplicon sequencing revealed the specific species composition of the bacterial community, thereby answering the basic question, who is in the community and in how many times they are repeating themselves, and if they are present, what they are doing basically. So in 2013, a powerful tool for predicting bacterial metabolic function called PICRUSt came into being, and related papers were published in the top journal Nature Biotechnology. Basically, PICRUSt abbreviation is for Phylogenetic Investigation of Communities by Reconstruction of Unobserved State, which investigate the communities through evolutionary reconstruction of unknown state. And this PICRUSt is also evolved from the lab of Curtis Huttenhover. The PICRUSt is work like this, you can see here, and it can also use the KEGG pathways. It's the combination of many input files, you can see here. General idea of PICRUSt. First, based on the full-length sequence of the 16S ribosomal RNA gene of the Meyer microbial genome, the gene functional spectrum of their common ancestor is inferred. Infer the gene infection profile of other untested species in the green gene database and construct gene function prediction profile for the entire spectrum of archaea and bacteria. Finally, the bacterial community composition obtained by sequencing is mapped into the database to predict the metabolic function of the bacterial community. How to implement specific step? We can see for the 16S ribosomal RNA gene sequence obtained by sequencing, perform close reference OTU picking and compare it with the green gene database to find the nearest neighbor of the reference sequence of each sequenced sequence and classify it as a reference OTU. Calibrate the obtained OTU appendix matrix according to the ribosomal RNA gene copy number of the nearest neighbor of the reference sequence, and based on the KEGG or egg energy and other gene functional spectrum data corresponding to the nearest neighbor of the reference sequence, the overall metabolic function of the bacterial group is converted and predicted. Now, in case 1, you can see the change in metabolic gene of corpse decomposers over time. We can see here the PCA plot, and we can see here further that PCOA analysis is usually performed and there is generally no difference in change over time. However, differences in time can be found by selecting metabolic related pathway. Select pathway related to corpse decomposition for fitting and display, such as nitrate to ammonia and cardivorine to petracine. In case 2, you can see different oral flora and related to function in smokers. This is the study of Wu Jing et al. With the title Cigarette, the Smoking and the Oral Microbiome in a large study of American adult. You can see here that there were significant difference in a total of 83 genes, functional metabolic pathway. Smoking greatly reduced the content of metabolic pathway such as carbohydrate and energy metabolism and xenobiotic degradation. Differential bacteria are related to different functions. So, how can we perform PICRUSt? There are ways we can perform under local server and we can perform online by using our ImageGP. So, what file need to be prepared? And first of all, compare green gene database to generate OTU table, which we already done in pipeline #9 number. Green gene database, as you know, is the oldest, most accurate, and most widely supported database. Based on its OTU table, function prediction PICRUSt, and morphological prediction, bug-based can be performed eventually. So, you can see OTU tab table which generated, it's look like this. So, method 1, we can generate some file of PICRUSt by uploading our OTU tab file on the ImageAPI, and then wait for a moment, we get the result. And when we get the result, we have a series of lists. And under the PICRUSt predict function result based on KEGG, we have 4 levels in total. Each level has originally and stem format result, you can see here. When we download all these files, and then we will run some commands in RStudio, and then we can analyze and visualize under STEM software. Method 2 is under Linux system. We can run PICRUSt environment and we can run these codes under Linux server one by one. So let's have a look. First of all, we just try to go back to our ImageAPI and open PICRUSt section. Let's see here, it's PICRUSt. come over here. We have the file, we come to our working directory, and now under result, see. Now see, we don't have here the folder directory of PICRUST, so we create the directory by ourselves, named PICRUST. So you just write PICRUST. Now, if you don't know how to use this, you can use demo data, and we want to paste our data here, and we need OTU tab text. So we will see where our OTU tab text is. We can see here, this is our OTU tab text. Let's go open it and have a look in Excel sheet. Paste it here. Now see, here is OTU ID. If there is a space between OTU ID, you have to finish this space and then select all, copy and paste over here. And then you can use default setting. You can use KEGG orthologs, and you can use text and biome. Let's see here. This is by default, it's now submit. We can see that our data is either okay or not. We can click this button, check data, and now see data format checked successfully. We can submit it now. And when we submit it, it will take some time to give us result. In method 1, we can use ImageGP to perform PICRUST analysis. Let's have a look. We already ImageGP open here. We can see here. We can click PICRUST. And then we have to copy our data and paste over here. So we will, this is our working directory, EasyAmplicon. We can see here. We can go to this results directory and open GG. Then we can see this is OTU tab text. Open it. And you can see here, this is our OTU ID and these are our samples. We can copy all and we will paste here. Now see, there is the gap between OTU and ID. We have to remove this space and bring it closer, and then copy all and paste here. When we paste here, after that we can see that our data is okay. Then we can click check data, and by default we will go with KEGG orthologs, and we will keep this text and biome setting and submit it. When we click submit, it takes some time to finish, and then we will get eventually a list of results generated via this one click. So preferably you can use PICRUST via ImageAP. When it generates some files, then we can download them and save under PICRUST directory, and then we can go back to RStudio and run the commands to proceed, and after that we can use STEM software to visualize them one by one. It takes some time to get it end, so wait patiently. Now this result we obtain, we can see here, PICRUST result, OTU biome table, NSTI sample summary result, PICRUST predict function result based on KEGG, PICRUST predict function result in function level 1, PICRUST predict function result in function level 2, PICRUST predict function result in function level 3. We can see here. So let's click PICRUST predict function result based on KEGG. it's have many result under this, like result table based on KEGG, result table based on KEGG, L2, L1, and it's generate file for stamp as well. Result table based on KEGG, KOL2, KOL3, KOL3, biome based on KOG, biome based on KOG, KOL1, so on. So what we're going to do, we just click one by one to each, and then we will save in our working directory. Let's do it. Base. And then we will see here. Let's see where we are. In the last, we did L2 biome. And then we will do. So after that, just copy all these and go to your working directory. Recently we created PICRUSt folder here, and we will save here all these files. When we paste here, then we have to do some modification with the names. We will delete this name number, and we just keep all level KO biome or level KO L1 biome like this. We will do one by one, because when we have to run in RStudio, it accepts the files with this all level KO or level KO L1. So we're going to delete this one by one. We can it's important to do here, otherwise you are in trouble when you execute the code in RStudio. So let's done with this right now. To avoid the error, we have to make sure that everything is we will done at right time. So we can now see this is the list of files we just got from ImageJP, and if you want to see the rest, you can see here we have OTU biome table here, like OTU biome table, we can click here, we generated here OTU biome, we start downloading here, normalize OTU biome table, we will get this. If you want to see NSTI sample summary result, just download these two files one by one. Okay. Now, in order to remove the confusion, we just delete all these files to make sure what we have and what we don't have. So in order to download further file, if you want to do OTU biome table, we can see here we have OTU biome table, we have normalized OTU biome table. Under NSTI sample summary result, we have NSTI sample summary result, NSTI sample summary result we have. And when we click here, PICRUSt predict function result in function level 1, we have here PICRUSt predict function result in function level 1 KO. We can see here, we have biome in function level 1, table of result of stamp analysis, result table in function level 1. When we click here, we have again PICRUSt predict function in function level 2. We can see here, you can see all these files we generated just by clicking once in ImageJP, and we got the results. So it is most one which is recommended to the users to follow this one. And if you want to do Linux server to perform PICRUSt analysis, you can do that one also if it's okay to you. So we can see, if you want to download them, you can download, and if you don't want to, then don't need it. And later if you see that you need some files, you just come here and you can start downloading the rest of the files one by one. Let's suppose we may need OTU biome table, so let's download it. Normalize OTU biome table. We can click this and this. Let's come over here. We can just copy all these and come over here under PICRUSt. We will paste it. Okay, do the same, remove the name which is not required, the numbers, delete it. Now see, we already have this file in the same location, so we don't need it. No. So what we're gonna do, we just delete them. So now we have all these files. Come back to the RStudio. Now see, under the section of feature prediction, which we can perform by PICRUSt, Fapratox, and bug-based bacterial phenotype. And under PICRUSt feature prediction, we're going to use PICRUST1, and optional methods are given here. Method 1 is utilizing ImageGP, the method 2 is Linux server, and under R language drawing, you can now, after generating file via using ImageGP, we can come back here and we can execute this code. And we see here it need L2, it need all level KO. Now we generated all these files. We can see all level KO, and we can see here what we need else. Mat text. So just do it and execute the code. Make sure that you are in your working directory, the EasyAmplicon. Here we are in the documents. So let's go back to because whenever you are not in your working directory, you have to go back and execute these 4 lines to come back in your working directory. Now see, we are here in the EasyAmplicon. So what we're gonna do, because here I can't see, so I will set directory here, our working directory, which is the EasyAmplicon. I click here and now see, come back to our terminal. So let's go back to the section of feature prediction. Okay, now we are here. Now we're going to run this code. You can see. We can see here under result, now we adjust our files L2 annotation text, L2 mat text, L2 SPF, and L2 text. Now in order to compare the differences, like let's suppose we take example of KO and WT. In order to perform the differential analysis, we can execute this code by, we have threshold here 0 and p-value 0.05, FDR 0.2, and the result is in the PICRUSt. Let's see and execute this code. Now deleted 11, enriched 7, and not significant 23. We can see here. We get here comparison text, KOWT text. And in order to generate a bar plot histogram for the specified group, we can execute this code, which will eventually give us mean bar PDF. You can see here it's generated KOWT mean A bar PDF. So in order to generate bar plot depicting significant differences between 2 groups, we will execute this code. Now we get here KOWT bar PDF. So let's have a look in our PDFs. Now see, here we have, you can see here KAGG pathways, and you can see here under cellular processes and genetic information, human disease, and this is the relative abundance. And you can see mean bar PDF here. So when you have generated all these files, we can use STAMP software as well to visualize them, and we can get more visualization result by using STAMP software. Let's have a look. We can click here. We can write here STAMP, run as administrator. So here we have our STAMP file as well. You can see here all level KOL1 STAMP text, all level KOL2 STAMP text, all level KOL3 STAMP text. So we can use it. Let's load data. Let's have a look how it will give us our results. And let's suppose we take all level KO L2 stem text, right? And then in the second we will use metadata text and click OK. Now as you click OK, you can see here we get the PCA plots. You can see here. Now if we change our group samples, you can see here, and we have multiple comparison plot here. You can have option, you can see p-value histogram. You can see here extended error bar. This is scatter plot. You can see here profile bar plot as well. So if you want to use group, you can come here and you can see one by one. This is sequence histogram. This is bar plot. This is extended error bar. And this is multiple comparison plot. And this is p-value histogram. So you can use any one which you want. We just change it to a function table and the experiment design remain unchanged. If PCA analysis is performed, the functional structure is similar to the bacterial group result and it's still clearly separated on the first axis. The specific function difference list as shown, you can see here in the picture. You can, for example, you can see here and you can see here as well. The specific function difference list is shown here. So what it depends that which analysis you want to perform, and according to your study and according to your research, you can choose any one of them. So you can use STEM software when you get the PICRUSt file, and then you can visualize under STEM. So come back to RStudio. Now PICRUSt 2, there is all, now we can see that we can perform PICRUSt 2 only under Linux server. So we can see here the code under Linux. In the appendix section, you can see you have in detail PICRUSt 2 codes giving under Linux environment. So you will run under the Windows subsystem Linux for Windows users. You come here and we will see here let's see here we have a section with the name of appendix in the pipeline. Now see here you have PICRUSt 2 environment export and import. You have PICRUSt 2 function prediction. So you need Linux system. You just only perform under this. You can't run in R, in Git Bash terminal. So this is how you can do PICRUSt analysis. So the next feature prediction we're going to discuss is looping through element of approx.